Welcome to another video for biology. In this video I'm going to be highlighting DNA, its structure, and its location. So let's first of all take a look at where this particular molecule is located at. So taking a trip down memory lane in biology, I just want to remind you of a few things. So I have highlighted here the, the plasma membrane of the cell. Okay, so if you recall the components of the cell way back when, in a previous biology video chapter. And then on the inside of the cell you have the cytoplasm. Okay, remember that's a gelatinous fluid where all the organelles pitter around. Okay, and speaking of the organelles, here we go. Okay, so we have the mitochondria. Okay, we have some peroxisomes, your Golgi complex, your endoplasmic reticulum. Okay, your smooth endoplasmic reticulum. Okay, so then what we're going to be highlighting in this video is your nucleus. To be more specific, oh, actually before I get more specific, let's look at the nucleus a little bit so we can get a closer look. Okay, so I have highlighted here your nuclear envelope. Okay, and then inside the nuclear envelope you have the nucleoplasm. This is where all the where your nucleolus and your chromosomes float in. And so your chromosomes are actually the ones that we will be taking a closer look at in this video. Okay, because in here, in these little X-shaped organelles or molecules, this is where all the genetic information for the human body lies. Okay, so this is where we're spending most of our time discussing in this video, is taking a closer look at these chromosomes. Okay, and looking at the DNA structure and what it does. So wrapped around all of those chromosomes is long, incredibly long strands of DNA. So you have about 3.7 trillion cells, I'm sorry, 37.2 trillion cells, and each one of those cells has a nucleus that is about 6 feet in length. Okay, so we're talking about quite a bit of information tightly wrapped around those chromosomes. In fact, the technical term for it is they're supercoiled inside of those chromosomes. And you'll notice that the DNA is wrapped around proteins called histones. So they're supercoiled, super tightly wrapped around those proteins, and they're bunched up in those little X shapes, your chromosomes. Okay, so let's take a look at what this DNA is actually made out of. Okay, the first molecule we're going to be looking at is a phosphate. Okay, deoxyribose. This is the sugar group. Okay, your sugar, and a nitrogen base. Okay, let's take a closer look at these nitrogen base. Why is it called a nitrogen base? Well, that's because it has nitrogen as one of its main atoms in this molecule. Okay, now just looking over again the three main components for deoxyribonucleic acid, DNA, is your phosphate, your sugar group, deoxyribose, and your nitrogen base. So looking at the nitrogen base with nitrogen as one of its main atoms, there are 4 types of nitrogen bases, okay, and they're paired up into 2 groups. The first pair is called your pyrimidines. These are thymine and cytosine. One of the best ways to remember this is thymine, cytosine, and pyrimidines all have Y in their spelling. And then the second pair is your purines. Okay, purines have rings. Okay, they have 2 rings. Right, there's your first ring and there's your second, and that is adenine and guanine. Okay, purines have rings and your pyrimidines have Y. Okay, so again, taking a look at all 4 of your nitrogen bases, pyrimidines and your purines. All right, so once again, we're going to keep reverting and reversing our thoughts. And so we're looking at the 3 main components to DNA: phosphates, your sugars, and then the nitrogen base, which are actually which comprise 4 individual ones. So let's take a look at how this looks put together. So here's a really small portion of the DNA molecule, and you probably can't really see much. It just looks like a bunch of circles and lines, and it doesn't really make a lot of sense. So what I'm going to do is I'm going to highlight in green and yellow the lateral aspects of this molecule so you can see them a little bit more clearly. Okay, so you can see the phosphate, sugar, phosphate, sugar. So on the outside of this ladder, okay, so picture DNA as being a ladder shaped. Okay, on the outside you have alternating molecules, a phosphate group, phosphate, sugar, and this just continues down the line, okay? So there you go. You can see it a little bit more clearly maybe with the inside rungs being faded. And again, that's the lateral aspect of the ladder, if you will, okay? So that's your sugar, phosphate alternating molecule. Now let's fade the outside and look at the actual rungs. This is the part of the ladder, if you will, where your hands and your feet go. Okay, these are where the nitrogen bases go. Okay, right now you probably can't really tell the nitrogen bases, but just to kind of help you out, so you can see here a single ring, and then you have a purine with two rings, and another purine, and another pyrimidine. Okay, so those make the rungs of the ladder. Okay, looking again, getting a closer look at how this molecule is positioned. Okay, and like I mentioned before, this pattern will continue down the line of this DNA molecule. All right, now, as I was mentioning before, I put it this way so that you can see what the molecule actually looks like in terms of its molecular structure. But what we're going to do is we're going to try and simplify this shape a little bit, and I'm going to replace each molecule with a shape and color. Okay, so I have there the different shapes that I will replace them with. Okay, so now we've removed our molecular structure. So this is what the molecule looks like with our new shapes in tow. So now getting rid of the molecular shape, we're going to see that it looks a little bit cleaner now. So this is why we're going to use this instead of our molecular shapes, because we can see a little bit better with what's going on in the DNA molecule here. All right, let's talk about nitrogen, your nitrogen base pairing. Okay, if you go back to the story of DNA, you'll remember Erwin Chargaff. He counted all the different number of times that these nitrogen bases came up, and remember, when he counted thymine, he noticed that thymine always equaled the number of adenine. Okay, and the same thing with cytosine, it would equal the number of guanine. Okay, so these particular nitrogen bases always had the same, almost exact proportions in any species. Okay, from the human, E. coli, any animal, any species, he always came to this particular truth that the DNA molecule, that these particular bases always proportion themselves out to be almost exactly the same. Okay? So for example, if I look at cytosine and I grouped it with adenine, and I counted the number of adenine the DNA molecule had, there was always an imbalance, so they didn't equal out. Okay? So this led Chargaff to hypothesize that perhaps these molecules, okay, these base pairings, perhaps they always paired up together in the DNA molecule, and in fact they did. Your A always went with T, adenine with thymine, and your guanine with cytosine. Just offhand, one of the probably quickest ways to memorize this, think of C and G. Those are the more curved letters. A and T have the more sharper angles to them. So just remember the letters with sharp angles go together, and the letters with the curvatures go together. And keep in mind as well that these base pairings, that they could also switch positions. It didn't matter which one was on the left or which one was on the right, but if T showed up, then it was always paired with a complementary base, which was A. Okay, and the same thing with G. If G came up, then it always paired up with C, and vice versa. So hopefully this makes a little bit more sense now what you're looking at. So you're looking at the lateral aspect of the ladder here with your phosphate, sugar alternating pattern on the outsides, on the most lateral aspect of this molecule. And then on the inside, the rungs, you have your base pairs. And this comprises the genetic code. Okay, this is, you can compare this to computer programming code like binary code. Think of all the websites and computer programs that you have are based off of a binary 0, 1, 0 repeating, randomly repeating pattern, and the two-number code codes for all the variety in computer programming that you see today. Well, likewise with DNA, you have these four base pairs, and that information codes for all the variety of characteristics that you see in the human race and in the animal kingdom. So it's pretty cool stuff. And and also, just still on the computer programming code, your binary code codes for two-dimensional information, and information codes for two-dimensional objects like something on the screen, whereas DNA, you have a two-dimensional code, which is a four-base pair that codes for 3D things, okay? Because human beings, animals are 3D objects, so you have a code that actually codes for constructing something that's got actual depth to it. So you'll notice here that I'm separating these two strands. DNA is a double-stranded, okay? It's a double-stranded molecule, bringing it back, and they're combined with, you see those little dotted lines? Those are hydrogen bonds. I will actually make a video explaining what is actually going on here with hydrogen bonds, okay? Now, pay attention to the sugar molecule on the lateral side, on the right side. Now, I have a dot there. That dot actually represents an oxygen molecule that is in that particular position. You'll notice that all the sugar molecules are pointing in one direction. They're pointing up. This is actually not accurate, okay? Now, the more accurate representation would be, if these sugar molecules on one end would actually be pointing down. So this is another one of the characteristics of DNA. So you have parallel backbones of alternating phosphate and sugar molecules, but on one end, on one side, they point down. This leads us to the next idea. So pointing up, right, sugar molecule, and the other one's pointing down. This is anti-parallel. Okay, anti-parallel. This idea was realized by Francis Crick, one of the main scientists that discovered DNA and its structure. Okay, so the anti aspect of this term is the fact that the sugar molecules are pointing in opposite directions. Okay, and again, I will make another video getting into even more detail as to what exactly does that mean to be anti-parallel. So anti, again, meaning the sugar molecules are pointing in separate and opposite directions, but yet they're parallel. So anti-parallel. So DNA is a double-stranded, anti-parallel double helix. So it's a ladder, but it's a ladder that's twisted. Okay? All right, so that does it for DNA, its structure, and location. Good luck in your studying.